Hello everyone, greetings for the day. Welcome to our live session today on the topic countercurrent mechanism. I am Dr. S.Kanimozhi, Professor of Physiology and the Course Director here at Dopamine. So this topic comes under the competency number 7.3. When we talk about countercurrent mechanism, this topic seems to be a little difficult topic, and for many students it is like a nightmare. At the same time, from an exam point of view, this is a very, very important topic repeatedly asked in examination. Not only from an examination standpoint, even for your better understanding of how the kidney concentrates urine, understanding of the physiological basis of this countercurrent mechanism is very, very important. The slides have been prepared in such a way that you can utilize the same content, answer or prepare your notes for your examination. And also I have tried to simplify it as much as possible so that you will have a very clear understanding of the topic. You don't have to memorize it again and again. Once you understand it, you will be able to write it with ease in your exam and also answer any question from countercurrent mechanism with confidence. So now let us move into our countercurrent mechanism. What are the learners expected to know at the end of the session? You should know the physiological basis and importance of countercurrent mechanism. What are the steps involved in the countercurrent mechanism? And finally, we will look at the physiological and pathological situation where the concentrating and the diluting ability of the kidneys get altered. Let us start by discussing the importance of countercurrent mechanism. So CCM stands for a countercurrent mechanism. So what is countercurrent mechanism? So this countercurrent mechanism is one with which the kidneys are able to form a concentrated urine. So if we look at the total body water, okay, so the total body water is controlled by two things. One is the fluid intake, the other is the renal excretion of water. Okay, so it makes sense, right? If we look at the homeostasis of total body water, that is controlled by two things. The amount of fluid we take in and also the amount of fluid that is excreted by the kidney. Now let us look at the amount of fluid that is excreted by the kidney. When we are talking about the renal excretion of water, that is again dependent on two things. What are those two things? One is the antidiuretic hormone, or also known as vasopressin. Where is this antidiuretic hormone released from? The antidiuretic hormone is released from the posterior pituitary. Second is the hyperosmolarity of the renal medullary interstitium. So in today's lecture, we are mainly going to concentrate on these two things with more importance given to this hyperosmolarity of the renal medullary interstitium. So to summarize, the total body water is controlled by the fluid which we intake or the renal excretion of water. When we are talking about the renal excretion of water, it is dependent on two factors. One is the antidiuretic hormone, the presence or absence of antidiuretic hormone. The second is the hyperosmolarity of the medullary interstitium. What is this interstitial space? So we know we have the capillaries. So outside the capillaries, you have these interstitial spaces and how the nutrient exchange keeps happening between these two. So when we are talking about interstitium, everywhere else in the body, the osmolarity of the interstitium is same to that of plasma. What is the osmolarity of the plasma? Plasma osmolarity is 300 milliosmoles per liter, right? So now this is the osmolarity of the plasma. The rest of the interstitium which is present in the human body, the osmolarity is the same. Except for the medullary interstitium. If you look at the medullary interstitium, the maximum osmolarity can go up to 1200 milliosmoles per liter. Okay, so that means one very unique feature what you have to remember is, among all the interstitiums that is present in the human body, it is only the interstitium that is present in the renal medulla which has hyperosmolar state. Okay, that is why we say renal medullary interstitium is very, very salty. So remember this one, you will be able to build upon this. The renal medullary interstitium is salty. The rest of the interstitiums in the human body, interstitial space, the interstitial fluid is isotonic to that of plasma. Okay. Now, what is a normal GFR? The normal GFR is around 180 liters per day. That means ideally, how much amount of urine are we supposed to excrete in a day? 180 liters, right? So since the GFR is around 180 liters per day, the volume of urine that needs to be excreted also is supposed to be 180 liters per day. Just imagine excreting 180 liters of urine per day. We would be almost doing only that task of sitting inside the washroom and excreting the urine. But does that happen? In real life scenario, do we excrete 180 liters per day? No. We only excrete a maximum of 1.5 liters per day. So if you see, normally the amount of urine excreted per day depends upon the fluid intake. But on an average, in normal conditions, a person excretes around 0.8 to 1.2 or maximum 1.5 liters of urine per day, which amounts to less than 1% of GFR. This is also an important MCQ. Okay, what percentage of GFR is normally excreted as urine? So what percentage of GFR is normally excreted as urine? Around 1.5 liters of GFR. Then why don't we excrete 180 liters per day? Because the GFR is 180, so ideally we are supposed to excrete 180 liters per day, but we don't do that. We excrete less than 1% of the GFR. Why? Because the kidneys have the capacity to pass urines with extreme osmolality. The kidneys have the capacity to pass urine with extreme osmolality without changing the solute excretion. So this is a very important point we have to note. Our kidneys are so specialized that depending upon the situation, it can either excrete a concentrated urine. If the osmolality is more, the urine is going to be highly concentrated, or it can also excrete a dilute urine. When it is excreting a dilute urine, the osmolality is less. That is why I have written it here. The kidneys have the capacity to pass urines with extremes of osmolality, depending upon the situation, without changing the solute excretion. Okay, so without changing the solute excretion, the kidneys can just move the fluid, either conserve water or eliminate water. Okay, and this function of the kidney, it forms the basis of volume and electrolyte homeostasis. Because of this capability only, the kidney is able to maintain the blood volume and electrolyte balance. Okay, now this ability of the kidney to concentrate urine forms the basis of countercurrent mechanism. Okay, so as I told you, the kidneys can excrete dilute urine as well as concentrated urine. So how much dilution it can happen and how much concentration can happen? So what is the maximum concentrating ability of the kidney? What is the maximum diluting ability of the kidney? Again, this is very important in your Viva as well as in MCQs. Okay, so let us look at it. Let us have a condition like diabetes insipidus. Okay, in diabetes insipidus, this antidiuretic hormone is absent. So please remember, what is the most important function of antidiuretic hormone? From the name itself, you know, antidiuretic hormone. So that means it is going to prevent diuresis. It is going to prevent the excretion of water in urine. So ADH inhibits the water loss in urine. Okay. So this antidiuretic hormone, in the presence of antidiuretic hormone, less amount of water will be excreted in the urine. Now, in diabetes insipidus, this antidiuretic hormone is absent. So now what is happening? Lot of water will be excreted in the urine. Okay, how much of water is excreted in the urine? Around 23 liters per day. And diabetes insipidus, 23 liters of water is excreted per day. What is the normal amount of urine that is excreted per day? Around 0.8 to maximum 1.5 liters per day. Compare this to diabetes insipidus. In diabetes insipidus, around 23 liters of urine is excreted per day, which amounts for around 12.5% of GFR. Normally, we excrete less than 1% of GFR. In diabetes insipidus, we excrete more than 12.7% of the GFR. When a person is excreting this much volume of urine, the osmolality is around 30 milliosmoles per kg of water. Okay, so the osmolality, the minimum osmolality that the kidneys can have, that is how much of dilute urine it can form. It can form around 30 milliosmoles per kg, which amounts to around 23 liters per day. So that is the diluting ability of the kidney. Let us look at the concentrating ability of the kidney when ADH is present. Example, in conditions like dehydration, vomiting, diarrhea, or the external environment when you are in a desert-like condition. So whatever may be the reason, when a person is dehydrated, the body wants to conserve the water. In dehydration, the body wants to conserve the water. So when you want to conserve the water, what is happening? The kidneys are producing a very high osmolarity. Okay, so here the osmolarity increases to 1400 milliosmoles per kg of water. When the kidney is concentrating so much, the amount of urine that is formed is around 0.5 liters per day. That means the maximum urine that can be formed is 23 liters per day. That is the diluting capacity of the kidney. The minimum amount of urine that is formed in a day is around 0.5 liters per day. Okay, when only 0.5 liters of urine is formed per day, if you look at the osmolality, it is around 1400 milliosmoles. When 23 liters of urine is formed in a day, then the osmolality of that urine is around 30 milliosmoles per kg. Why is this level 0.5 liters the minimum urine that can be formed? Because in the human body, for the normal survival of the human being, at least 600 milliosmoles per kg of water of solute has to be excreted. The solute load, minimum solute load that needs to be excreted out per day is around 600 milliosmoles. So if you want to excrete 600 milliosmoles of solute, then at least 0.5 liters of urine has to be formed. Okay, so now I hope you are very clear about this ability, concentrating and diluting abilities, extremes of concentrating and diluting abilities in the kidney. Okay, so thus osmolality of urine is a very important index in the assessing the diluting and concentrating capacity of the kidney. Again, this is a very important MCQ. Okay, which is the index that helps us to know whether the kidneys are producing a dilute urine or concentrated urine. So this is again a very important MCQ. Osmolality of the urine is an index of the concentrating and diluting capacity of the kidneys. Now, when we are talking, already we have been talking about the same thing, right? Antidiuretic hormone and the concentrating ability of the kidney. So when we look at it, the antidiuretic hormone is an external factor which plays an important role in urine concentration and dilution. That is, if antidiuretic hormone is present, the concentration of the urine happens. If antidiuretic hormone is not there, the reabsorption of water doesn't happen, so a dilute urine is formed. So antidiuretic hormone acts as an external factor, whereas the kidney has its own intrinsic mechanism. The kidney has its own intrinsic mechanism through which it concentrates its urine both in normal as well as in abnormal condition. So that inherent mechanism that is available in the kidney for concentrating the urine is called as the countercurrent mechanism. Now I welcome you to the discussion on countercurrent mechanism. Now only we are starting with the actual discussion on countercurrent mechanism. So this countercurrent mechanism, when they ask you as an essay question, I would suggest you approach the question in under these subheadings. So first we will discuss the definition of countercurrent mechanism. Then we will look at the components of countercurrent mechanism, how it happens, what are all the steps that involved in the countercurrent mechanism. Finally, we will look on what are the factors that affect the countercurrent mechanism, what are the functions of countercurrent mechanism, what is its role. By now you know it is very clear. What is the most important role? Forming a concentrated urine, yes. And then what are the other systems where such countercurrent mechanism is present in the human body? Finally, we will touch upon the applied aspects. So let us go one after the other. Now let us see the definition of countercurrent mechanism. What is the definition of countercurrent mechanism? So this countercurrent, current means what? Direction of flow. Countercurrent means what? The direction should be opposite to one another. So a countercurrent system refers to a system in which the inflow runs parallel to, so the first point is inflow runs parallel to, counter to, and in close proximity to the outflow. Okay, so these three things has to be satisfied. For a system to be called as a countercurrent system, we need three things that needs to be satisfied. One is the system should have two tubes that are parallel to each other. So you should have two tubes that are parallel to each other. the flow in this tube has to be counter to one another. That is, in one tube, if the flow is towards the inside, the other tube, the flow has to be on the outside. So, we need two tubes, two parallel tubes. The flow needs to be counter to one another, and also these tubes have to be in close proximity to each other for some distance. And another very important point is the membrane needs to be selectively permeable. Okay, so the fourth another important point is selective permeability. When all these criteria is met, then we call that system to be a countercurrent system. Now, where in the kidney, where in the renal tubule, where in the nephron are we able to find such a system? Yes, you're right. So, it is your, what is it? It is your loop of Henle. So, it is the loop of Henle where we see this countercurrent system existing. How? So, what is this portion? So, we have the glomerulus. From the glomerulus, we have the proximal convoluted tubule. Okay. Then from the PCT, you have the descending limb of loop of Henle. So, first is your descending limb of loop of Henle. Then we have the ascending limb of loop of Henle. So, this is your ascending limb of loop of Henle. Okay. So, this descending limb and ascending limb is forming this part 1. That is, two parallel tubes are there. Then, they are running close to each other. Okay. And the membrane of this is semi-permeable. And another very important is the inflow is opposite to the outflow. So, in our descending limb, the fluid is coming towards the medulla. In the ascending limb, from the medulla, the fluid is going out towards the cortex. So, all the criteria is matched by our loop of Henle. So, thus, the countercurrent mechanism in the nephron exists in the loop of Henle. Okay, so I hope the definition of countercurrent is clear. Now, let us look at the components of countercurrent system. What are all the components of countercurrent system? Classically, if you look at your textbook, they would have given two components. I have added another component as well because this also has a very important role. So, what are the two important components of countercurrent system? We have the countercurrent multiplier, we have the countercurrent exchanger. We also have one more component, which is the osmotic equilibrating device. Okay, what is it? Osmotic equilibrating device. Later, we'll be discussing what is this osmotic equilibrating device. For now, remember the countercurrent has three components. The first component is a countercurrent multiplier. So, what in the loop of Henle acts as a countercurrent multiplier? It is the loop of Henle actually acts as the countercurrent multiplier. What does it do? It produces an hyperosmolar medullary interstitium. And this medullary interstitium hyperosmolarity is produced in a gradient. Okay, the hyperosmolarity that is produced in the medullary interstitium follows a gradient. It is not one single value throughout the medullary interstitium. So, slowly gradient means what? It keeps on increasing. So, it is the loop of Henle which is responsible for producing a hyperosmolar medullary interstitium, and also that hyperosmolarity is produced in gradient. Okay, so that is why the countercurrent multiplier is nothing but the loop of Henle. Multiplication is a function of the loop of Henle. Then the second component is a countercurrent exchanger. Okay, this countercurrent exchanger function is done by the vasa recta. So, the vasa recta is the countercurrent exchanger. And what is it doing? It is responsible for maintaining the gradient and the hyperosmolarity that has been already established by the multiplier. The multiplier, that is a loop of Henle, establishes the medullary osmolarity and its gradient. The vasa recta helps in maintaining it. Okay, so the vasa recta maintains the gradient that has already been established by our loop of Henle. Then we have the third one, that is the osmotic equilibrating device, which is nothing but the function of the collecting duct. Okay, the collecting duct, which is juxtaposed to the loop of Henle and vasa recta, acts as the osmotic equilibrating device. Okay, what do you mean by osmotic equilibrating device? It helps to maintain an osmotic equilibrium on either side of the cell membrane, that is on either side of the collecting duct, between the medullary interstitium and the fluid in the collecting duct. How it does, we will see shortly. So, okay, so thus the components of countercurrent mechanisms is clear. So, we have the countercurrent multiplier, which is the function of the loop of Henle, countercurrent exchanger, which is the function of the vasa recta, and the osmotic equilibrating device, which is the function of the collecting duct. With this in mind, let us move on to the next part. Okay, so here this is a pictorial representation of the different parts of the countercurrent mechanism. So the structure, what is the structure? Now you will help me to label this. What is the structure here inside? This is your, what is this portion? This is our loop of Henle. Okay, this is a loop of Henle, which is our countercurrent multiplier. So this green structure is our loop of Henle, which is the countercurrent multiplier. So what do you see here? This portion is the descending limb of loop of Henle. So how is the flow of the filtrate? From the proximal convoluted tubule, it is coming down. The flow is in the downward direction. Then we have, this is what is the, from the descending limb of loop of Henle, now you have the ascending limb of loop of Henle. If you look at the ascending limb of loop of Henle, it has two portions. So this is the thin ascending limb of loop of Henle, and this region is your thick ascending limb of loop of Henle. Okay, so descending limb of loop of Henle is only one, thin descending limb of loop of Henle. Whereas when it's ascending up, we have two portions, thin ascending limb of loop of Henle, thick ascending limb of loop of Henle. Okay, so this forms the countercurrent multiplier. Then closely surrounding our loop of Henle, we have a blood vessel, a capillary, which is called as the vasa recta. The vasa recta, as I already told you, forms the countercurrent exchanger. What I want you to see here is, look at the flow. Okay, so this is the descending limb of loop of Henle, but when you look at the vasa recta, okay, so this is the vasa recta. When you look at the vasa recta, this is the ascending limb of vasa recta. Okay, this is the ascending limb of the vasa recta. Look at the direction of the flow. It is opposite to the direction of the flow in the loop of Henle. Now look at this portion. What is this portion? This is the arrow is coming down. So this is the descending limb of vasa recta. Okay. So essentially what I want you to remember here or understand here is, in the loop of any countercurrent mechanism, we have two parallel tubes, and the flow in the tubes have to be opposite to each other. Okay. So this is our loop of Henle. This is the descending limb, this is the ascending limb. In the descending limb, the flow is in this direction. In the ascending limb, the flow is in the outward direction. Okay. Then we have the vasa recta, which closely surrounds and mimics the loop of Henle. It is closely surrounding the loop of Henle. In vasa recta, the direction of flow is opposite to that of our loop of Henle. Okay, so that means this is the descending limb of vasa recta. The flow is down, and this portion is the ascending limb of vasa recta. So the flow is up. So this concept have to be very, very clear. Okay, I hope it is not confusing. Please take a time to understand this point. Only when you understand this point, when we move further, it will be easier for you to grasp the concept. Okay, so that is with regards to the loop of Henle and the vasa recta. Now let us look at the collecting duct. This collecting duct, I told you, acts as a osmotic, everywhere it is written, osmotic equilibrating device. What do you mean by osmotic equilibrating device? It will try to maintain an osmotic equilibrium on either side. That means the inside you have the filtrate, outside this portion is the medullary interstitium. All this region, what I am writing is MI is the medullary interstitium, right? So now this vasa recta will try to equilibrate with the medullary interstitium. It will try to bring an osmotic equilibrium. Okay, so that is why it is called as osmotic equilibrating device. Okay, let us discuss in detail regarding the concept of medullary interstitial hyperosmolarity. Why are we discussing this medullary interstitial hyperosmolarity? Because by now it is very, very clear that the countercurrent mechanism is responsible for the kidney's ability to concentrate urine is because of the countercurrent mechanism. How is the countercurrent mechanism able to concentrate the urine? Because it has the capacity to make the medullary interstitium hyperosmolar. As I told in the starting, nowhere else in the human body the interstitial fluid is hyperosmolar. Everywhere it is isotonic to that of the plasma. It is around 300 milliosmoles per liter. Whereas it is the medullary interstitium which is actually having the capability to become hyperosmolar. Okay, so how the medullary interstitium becomes hyperosmolar? That function is done by a loop of Henle. Now I think it is clear your each point is fitting into the other, right? So we want only if the medullary interstitium is hyperosmolar, the kidneys will be able to concentrate the urine. And this function is known as the countercurrent mechanism. To have a hyperosmolar medullary interstitium, we have a structure called as a loop of Henle, which actually increases this osmolarity of the medullary interstitium. Okay, so thus the interstitial fluid of the medulla is very, very important in concentrating the urine. So that is the meaning of this line. The interstitial fluid of the medulla is critically important in concentrating the urine. Why? Because this interstitial fluid, this medullary interstitium, the fluid of this medullary interstitium, only if it is hyperosmolar, it will drive the reabsorption of water. Okay, I will tell you again. This portion is the medullary interstitium. So here what do you have? We have the loop of Henle, right? In the loop of, this is your loop of Henle. Inside the loop of Henle, what do we have? The filtrate. Okay. Now, what is our aim? What is our goal? To make this region hyperosmolar. We have to maintain this as hyperosmolar. See, surrounding the nephron, everywhere you have the medullary interstitium. Our idea is to make the medullary interstitium hyperosmolar. So, how? If you want to make the medullary interstitial fluid hyperosmolar, what should you do? You should add solutes or you can remove fluid. By doing either one of this, either you add solute or you reabsorb the fluid. By doing this only, it is going to make this interstitium hyperosmolar. So, what the kidneys will do is, in this descending limb of loop of Henle, water will be reabsorbed. Okay. And this medullary interstitium hyperosmolarity acts as a driving force for reabsorption of water. That is the meaning of this line. Okay. Now, as I told you, the normal osmolarity of the plasma and other body fluids is only 300 milliosmoles per kg of water. Whereas, the interstitial fluid in your medulla can go up to 1200 milliosmoles per liter. Whereas, if you look at the cortex, okay, if you look at the renal cortex also, the medullary interstitial osmolality is only 300 milliosmoles per kg of water. Okay. So now I told, so one point is very clear, that the medullary interstitium will become hyperosmolar. The second point what we discussed was, this hyperosmolarity is established in a gradient. So this is what you see here is your medullary interstitium. This is the outer cortex. This is the inner medulla. Okay. As I told, in the outer cortex, it is same as plasma. The interstitial osmolarity is same as plasma. Okay. As you keep coming down, as we keep coming down into the medulla, can you see the interstitial osmolarity keeps increasing from 300, it goes to 400, then 600, then 800. Finally, at the tip, it is the highest, around 1200 milliosmoles. Okay. So, the osmolarity of the renal medulla is higher than that of the plasma. Already I told you, okay. And this goes on increasing from 300 to 1200 at the tip where you have the renal papillae. So, this is called as the medullary gradient. Now, do you understand the concept of medullary gradient? As we keep going down into the renal medulla, the osmolality keeps on increasing. That is called as medullary gradient. Okay. Now, let us look at the special characteristics of loop of Henle that causes the solutes to be trapped in the renal medulla. Okay, so by now you are very, very clear. This concept is very clear that it is the loop of Henle which increases the medullary interstitial hyperosmolarity. And I already told you, to increase the medullary interstitial hyperosmolarity, what needs to be done? Solute needs to be added and also water needs to be reabsorbed. When you remove the fluid and when the solute keeps on adding, what will happen? Automatically that region is going to become hyperosmolar. So, this hyperosmolarity is done by the loop of Henle, and there are certain specialized characteristics that the loop of Henle possesses which leads to this increase in hyperosmolarity. What is it? Let us start looking from here. So you have the proximal convoluted tubule. Then from the proximal convoluted tubule, what do you have here? We have the descending limb of loop of Henle. Please remember, this descending limb of loop of Henle, impermeable to solute. Okay. Permeable to water. Okay. So this is the most important point you have to remember. First point is the descending loop of Henle is impermeable to solute, but it is permeable to water. So what do you understand by this? That means from this portion, the filtrate which is present in the descending limb of loop of Henle, from this fluid only water will leave. Okay. Only water can leave from the filtrate that is present at the descending limb of loop of Henle. The water from here, where will it go? Obviously, the water is going to go into the medullary interstitium. Fine. This is one special characteristic. The second special characteristic, this is a descending limb. What is here? You see here, this is the ascending limb of loop of Henle. This ascending limb of loop of Henle has two portions. This is the thin portion. This is the thick portion of the ascending limb of loop of Henle. Be it the thin or the thick ascending limb of loop of Henle, it is permeable to solutes. It is permeable to solutes. If it is permeable to solutes, then it is impermeable to water. Okay. It does not allow water. Okay. This does not allow solute. So, the ascending limb of loop of Henle is impermeable to water. It is permeable to solute. That means, what do you understand? From this portion, the filtrate that is present in the ascending limb of loop of Henle, from there only the solute will move out. The water will stay. Okay. That is why you are seeing an arrow here. From here, sodium chloride. What is sodium chloride? Basically solute. The sodium chloride is coming outside. So, this is the second property. Now, coming to the third important property, what you have to remember, or special characteristic, what you have to remember. What is this portion? This is the collecting duct. This is the cortical collecting duct. This is the medullary collecting duct. Okay. The cortical collecting duct is impermeable to urea. Whereas, look here, the medullary collecting duct is permeable to urea. Okay. So, when we are looking at, we finished looking at the loop of Henle. Now, we have moved on to the collecting duct. So, the cortical portion, the portion of the collecting duct that remains in the cortex is impermeable to urea, whereas the medullary portion of the collecting duct, from here we have the medulla, right? The medullary portion of the collecting duct is permeable to urea. Even the outer medullary portion is not so much permeable to urea. It is only the inner medullary. The cortical collecting duct is impermeable to urea. The outer medullary collecting duct is also not so much permeable to urea. It is only the inner medullary collecting duct which is permeable to urea. Another point about the collecting duct is, if antidiuretic hormone is present, then it becomes, or both the cortical as well as medullary collecting duct becomes permeable to, both are permeable to water. If ADH is present, if antidiuretic hormone is present, they become permeable to water. If antidiuretic hormone is not there, they are not permeable to water. Okay. So, these are the special characteristics that we have to keep in mind. Only when you remember these special characteristics, we will be able to understand the steps of countercurrent mechanism easily. Okay. Now, let us look at what are all the major factors that contribute to the solute concentration in the renal medulla. Why are we talking about the factors that contribute to the solute concentration in the renal medulla? Only what is our ultimate goal? To increase the interstitial solute content. That means then only the interstitium is going to become hyperosmolar. Now, we are looking at 4 to 5 important factors which is actually increasing the solute concentration in the medullary interstitium. So, what are those things that increases the solute concentration in the medullary interstitium? The first one, and the most important is the active transport of sodium and co-transport of potassium and chloride and other ions out of the thick portion of the ascending limb of loop of Henle. What do you understand by this? Let me tell you here. This is the first factor. What is this portion? First, you should be able to identify. What is this portion? This portion is the thick ascending limb of loop of Henle. Okay. So, in this thick ascending limb of loop of Henle, you have a transporter. Name of this transporter is sodium potassium 2-chloride transporter. Okay. This is an active mechanism. Active mechanism means what? Needs energy, ATP. So, where is this active transporter present? This active transporter is present in the thick ascending limb of loop of Henle. Please remember this thick portion of the ascending limb of loop of Henle has this sodium potassium 2-chloride co-transporter. This is an active mechanism. Okay. Because of this, what is happening? Sodium, potassium, chloride, all these ions are, what is this region I told you? Medullary interstitium. So, all these ions are getting transported into the medullary interstitium against their concentration gradient because it is an active mechanism. So, now what will happen when you put solute into the interstitium? What will happen to the osmolarity of the interstitium? Obviously, the interstitial osmolarity will increase. Okay. This is the first factor. Then we will move on to the second factor. What is the second factor? Second factor is active transport of ions from the collecting duct into the medullary interstitium. So, what is this portion? This is the collecting duct, right? In the collecting duct also, can you see here sodium chloride? This is an active pump. Mainly, which is the pump here? Sodium, potassium, ATPase pump. Okay. So, now in the collecting duct, we have the sodium potassium ATPase pump which actively transports 3 sodium into the medullary interstitium. So, now again in the medullary interstitium, what is happening? The solute load is increasing. Already we saw step 1, the thick ascending portion of the loop of Henle. This is the step 1 where solutes are added into the medullary. All this region you see in white is the medullary interstitium. So, now already active transporter present in the thick ascending limb of loop of Henle has transported ions into the medullary interstitium, making it hyperosmolar. Additionally, now what is happening? In the collecting duct also you have the sodium potassium ATPase pump which is also pumping ions into the medullary interstitium. So, what will happen? The interstitial osmolarity increases. Then, third factor which increases the interstitial osmolarity is the passive reabsorption of sodium and chloride from the thin ascending limb of loop of Henle which is also impermeable to water. So, this is the third point. Okay. So, the first point was here. The second point we discussed was in the collecting duct. Now, we are coming to the third point which is the thin ascending limb of loop of Henle. So, in this thin ascending limb of loop of Henle also you see the solute is going into the medullary interstitium. Can you see? Sodium chloride is getting deposited into the medullary interstitium. The only difference between 1 and 3 is here it was an active mechanism. Okay. Here it is a passive mechanism. Okay. Here it is a passive mechanism through which diffusion, so passive reabsorption, diffusion of sodium chloride ions from the fluid, from the filtrate that is present in the ascending limb of loop of Henle into the medullary interstitium. So, this is also going to, so ultimately in each of these points, what are we seeing? The medullary interstitium is getting loaded with solute. Okay, so this is also contributing to the solute load of medullary interstitium. Okay, why so much things are depositing the solute? Because normally the interstitial molarity is around 300. Now we have to increase it to up to 1200 milliosmoles, right? So we have to increase it to 1200 milliosmoles. So for that only solute is continuously getting added from different ways. What is the fourth factor that increases the medullary interstitial hyperosmolarity? That is the facilitated diffusion of large amounts of urea from the inner medullary collecting duct. So what is this region? This is the inner medulla. So in the inner medullary collecting duct, what do you have? You have the urea. Okay. So urea, this is again a passive process. This is a passive mechanism by which urea is getting added into the medullary interstitium. Again, urea is a solute. So what it is going to do? It is going to increase the medullary interstitial hyperosmolarity. What you have to remember is, this is a passive process. This is not an active process. Okay. Finally, if you see, when we look at the medullary interstitium, in the cortical, in the collecting duct, in the medullary, inner medullary collecting duct, if you see, already sodium chloride with the help of sodium potassium ATPase pump actively enters into the medullary interstitium. Then passively urea is also entering into the, this is passive, this is an active process. Okay. Then passively urea is also entering into the medullary interstitium. Then when I told, what did I say? Collecting duct is an osmotic equilibrating device. Okay. So collecting duct is an osmotic equilibrating device. That means what it will do? It will try to maintain the osmotic equilibrium on both sides. So now what is happening? Solutes have left the filtrate in the collecting duct, and the concentration of solute is more on the medullary interstitium compared to the lumen of the collecting duct. So automatically what will happen? Water is leaving the lumen. Okay, so water will also leave the lumen and it will enter into the medullary interstitium. But if you see, only small amounts of water, this is very important. Diffusion of only small amounts of water from the medullary tubules into the interstitium will happen. This is far less than the reabsorption of solute. Okay, compared to the solute, so much of urea has entered into the medullary interstitium, so much of sodium chloride has entered into the medullary interstitium. Compared to this, the amount of water that enters the medullary interstitium is very, very minimal. It is less. Okay, so when less water is entering into it, that means the medullary interstitium will get diluted very minimally. So it is able to have higher interstitial osmolarity. Okay, so these are the 5 important factors which actually increases the medullary interstitial hyperosmolarity. In the coming slides, we are going to see how these factors happen step by step. Okay, so for now, I will just give you a brief overview so that you stay on track. So we have around 5 important factors which increases the medullary interstitial hyperosmolarity. First point is, why should you increase the medullary interstitial hyperosmolarity? We have to increase only if the medullary interstitium becomes hyperosmolar, the kidneys will be able to form a concentrated urine. Okay, so that is why we want to increase the medullary interstitium osmolarity. Okay, how does it happen? First point. First for that is the active transport of sodium chloride, sodium, potassium and 2 chloride. Okay, from the medullary, from the ascending limb. Okay, ascending limb of the thick ascending limb of loop of Henle into the medullary interstitium. Okay, then what happens? Second, passive. This is passive. Okay, what is happening? Passive transport of solutes like sodium and chloride from the thin ascending limb of loop of Henle into the medullary interstitium. So this is also increasing the interstitial hyperosmolarity. Third thing, what is happening here? In your medullary collecting duct, active transport of again sodium and chloride with the help of sodium potassium ATPase pump into the interstitial lumen, into the medullary interstitial fluid. You have the sodium chloride which is entering, increasing the medullary interstitial osmolarity. Fourth, urea. Urea is also from the same portion, from the medullary collecting duct. This is passive. Passive movement of urea from the fluid that is present in the collecting duct. That fluid is nothing but the filtrate which is ultimately going to form the urine. So from here, passive movement of urea into the interstitium, that is also increasing the interstitial osmolarity. And finally, very little movement of water, comparatively less movement of water happens from the medullary collecting duct into the interstitium. So all this collectively will increase the solute load in the renal medulla. Okay. Now let us look step by step how it is increasing. What are all the steps involved in causing the hyperosmotic renal medullary interstitium? Okay. First step, what you see here is What is this portion called as? Proximal convoluted tubule. Okay. From the glomerulus, the fluid, the filtrate enters into the proximal convoluted tubule. Okay. Most of the 100% of glucose, other nutrients like amino acid, more than 65% of the ions and water, everything is reabsorbed from the proximal convoluted tubule. Now, after reabsorption, including urea, after reabsorption of all these substances, if you look at the osmolarity of the filtrate or the fluid that is present in the proximal convoluted tubule, it is around 300 milliosmoles. Why it is 300? Because it is isotonic to that of plasma. It is isotonic to plasma, which is around 300 milliosmoles. Okay. So that is why the first step is assume that the loop of Henle is filled with a concentration of 300 milliosmoles per liter of fluid. So from the PCT, okay, now do you know why it is 300 milliosmoles? Actually, it is 290 milliosmoles. We have just rounding it up for ease of understanding. Okay, so from the proximal convoluted tubule, we have a fluid that is isotonic to that of plasma entering into the loop of Henle. So what is this portion of the loop of Henle? Descending limb of loop of Henle. Okay, then we have the thin ascending limb of loop of Henle, thick ascending limb of loop of Henle. Okay, so now in step 1, we have a filtrate or fluid coming from the proximal convoluted tubule and filling the loop of Henle. Now, if you look at the osmolarity, it is around 300 milliosmoles. Now, you know why it is 300 milliosmoles. Okay, and then what is happening? This is the medullary interstitium. The medullary interstitium is also having an osmolarity of 300 milliosmoles. Why? Because it is trying to equilibrate on both the sides. So, that is the first step. Second step, what is happening? Now, you remember those 5 factors I told you. We discussed those 5 factors which increases the medullary interstitial hyperosmolarity. Please remember the first factor. What was the first factor I told you? Active ion pump. Can you just recall and tell me what is the name of the active ion pump? It is the sodium potassium 2 chloride pump. Okay. So we have the sodium potassium 2 chloride pump, and if you can remember, where is this pump present? In the thick, please be very clear, it is in the thick ascending limb of loop of Henle. Now this pump, so this is the same pump I am talking. Okay. So that first factor, that pump is going to pump from where to where? From this is the medullary interstitium, this is the fluid that is present in the loop of Henle. This portion is the descending limb, this is the ascending limb. Okay. So ascending limb of loop of Henle, we have this pump which is going to pump solutes from here to here. So obviously the medullary interstitial is becoming more. Can you compare from this? Initially when we started, the medullary interstitium was only 300 milliosmoles. Now because this pump has pumped solutes into the medullary interstitium, now it has increased to 400 milliosmoles per liter. Okay. Now look at the difference between here and here. What is this portion? This is a portion which is containing the filtrate. Okay. Or we can even call it as urine. So from here, if you compare to the medullary interstitium, what is the difference? 200 milliosmoles. That means this sodium potassium 2 chloride pump has a capacity to establish a concentration gradient of around 200 milliosmoles per liter. This is again a very important MCQ as well as Viva question. What is the capacity or capability of that active pump that is located in the thick ascending limb of loop of Henle? That is 200 milliosmoles. It can establish a gradient of around 200 milliosmoles. Okay. So now as a result of this pump, what has happened? The fluid inside the renal tubule, that is the thick ascending limb of loop of Henle, what is happening? This has become hypotonic. Why this fluid has become hypotonic? Because it has lost solutes and the interstitium has become hypertonic. Because of this pump, this has happened. Now what is this? Is a step 2. Step 3, what is happening? So now you know how we got this number 400, right? Now what is happening? Tubular fluid in the descending limb of loop of Henle. So this is the descending limb of loop of Henle. Now as a result of that active pump, what is happening? the medullary interstitium, the solute load has increased. Okay. The kidneys is now trying to maintain an equilibrium. It tries to reach a osmotic equilibrium. Okay. This side is only permeable to solutes. Only solutes move. The descending limb, only water moves. From this side only water will move. So how is it going to? From this side solute has gone. Okay. See, if you can compare here, since this portion is permeable only to solute, the active pump has pumped solute out. Now to maintain an equilibrium, equal amount of solvent, equal amount of water also has to move. But we know the thick ascending limb of loop of Henle is impermeable to water. Okay, so here only solutes can move, water will not move, impermeable to water. So now what is happening? The descending limb of loop of Henle, is permeable only to water. So automatically to maintain an osmotic equilibrium, what is happening? Water from the descending limb of loop of Henle will move into the interstitium. When you see dotted line, that means what? Passive movement. Dotted line means passive movement. So passive movement of water. So this is also, if you read your books, they will say passive reabsorption of water from the descending limb of loop of Henle into the medullary interstitium. That means water is moving from the fluid in the descending limb of loop of Henle into the medullary interstitium. Now you tell me this portion is losing solvent, fluid is lost. So what will happen to the osmolarity? Now the solute will increase here, because as the fluid is leaving, it is getting more concentrated. When it is getting more concentrated, what do you call it as? Hypertonic. So that means the descending limb of loop of Henle, the fluid, as the descending limb of fluid comes down the descending limb of loop of Henle, it is going to become hypertonic. Okay, fine. So that is the third step. Fourth step. Now what has happened? One round is over. Now additional fluid from the loop, additional fluid will flow from the proximal convoluted tubule into the loop of Henle. See, at one point, at any point of time, please remember, at any point of time, only 25% of the filtered fluid from the glomerulus will enter into the loop of Henle. Okay, 25% of the filtered fluid will enter. Okay, so in the first go, first 25% of the filter fluid has entered and it has completely traversed the descending as well as the ascending limb of loop of Henle. Now next the cycle repeats. Now the additional flow of fluid into the loop of Henle from the proximal tubule will happen. Okay, so now again a new batch of fluid is coming from the PCT. It enters into the descending limb of loop of Henle. Okay, now what will this do? This causes the hyperosmotic fluid previously formed in the descending limb to move to the ascending limb. Can you see? This fluid is coming and pushing. We are calling it as previously formed hyperosmotic fluid. Already I told you why it is becoming hyperosmotic. Why it became hyperosmotic? How this number became 400? Because it lost water. Okay, water was reabsorbed from here, from the fluid in the descending limb of loop of Henle into the medullary interstitium. Water moved here. That is why it became hyperosmotic. Now this hyperosmotic fluid is pushed into the ascending limb of loop of Henle. So this fluid is getting pushed. As this is getting pushed into the ascending limb, what is happening? The hypotonic fluid that was present in the ascending limb is going towards the distal convoluted tubule. Okay, so that is the fourth step. Now coming to the fifth step. In fifth step, what is happening? The same process only. Once this fluid is in the ascending limb of loop of Henle, the one which got pushed, right? Once it reaches in the ascending limb of loop of Henle, what is happening? We have the transporter. Now you please tell me the name of this transporter. Yes, sodium potassium 2-chloride transporter. This is an active transporter. What is it going to do? It is going to transport solutes in the form of sodium, potassium and chloride from the collecting duct, from the loop of Henle into the medullary interstitium. Okay. Now, why do you see, and what is the maximum difference it can produce? A difference of around 200 milliosmoles, 500 minus 300. So, you have a difference of around 200 milliosmoles per liter. Okay. So, once the fluid is in the ascending limb, additional ions are pumped into the interstitium. Okay. That is why you see, when we started, if you remember, the medullary interstitial osmolarity was 300. Now, it has become 500. Because one round of fluid was over, the another fresh batch of fluid from the proximal convoluted tubule came into the descending limb of loop of Henle, which got pushed and entered into the ascending limb. Once this fresh batch entered the ascending limb, again the active pump is transporting it into the interstitium. So now you just imagine, from 300 we have already reached 500, right? Just in 2 cycles we have reached 500 milliosmoles per liter. What is the 6th step? Now what is happening? The fluid in the descending limb reaches equilibrium with the same step is getting repeated. Okay. 6th is the same step. So what is happening? Now from here ions are pumped into it. What will happen? In the descending limb of loop of Henle, what will happen? Water. Okay. Water will move from the descending limb of loop of Henle into the medullary interstitium. So that is the 6th step to maintain the osmotic equilibrium. Okay. Now, these steps are repeated several times over and over. So, all those steps I told you, mainly step 1, 2 and 3. So, these steps will be repeated over and over again, finally adding a lot of solute into the medullary interstitium. Steps are repeated over and over with the net effect of adding more solute to the medullary interstitium in excess of the water. Okay, and when it will stop? When it reaches the maximum gradient of around 200 milliosmoles per liter. Okay. Do the kidney always establish a medullary interstitial osmolarity of 1200? No, not always, depending upon the situation. If the kidney wants to conserve water, and depending upon the degree of conservation it wants to do, that is depending upon the degree of dehydration only, the medullary interstitial hyperosmolarity gradient will also get established. If it wants to conserve only very little water, then it won't go to the maximum of 1200. It will stop at the level of how much water it wants to conserve. Okay. So, those are the most important steps that leads to increase in medullary interstitial hyperosmolarity. So, thus to summarize, the repetitive reabsorption of sodium chloride in the thick ascending limb of loop of Henle. So, this is very, very important. So, which is the most important portion of the nephron that is responsible for the countercurrent mechanism? It is mainly the thick ascending limb of loop of Henle. So, the repetitive reabsorption of sodium chloride in the thick ascending limb of loop of Henle and the continued flow of new sodium chloride from the proximal tubule leads to a mechanism called as countercurrent multiplier. Okay. So, this is with respect to countercurrent multiplier. So, all those 7 steps which we saw is summarized here in this one picture. Okay. So, we have the thick ascending limb of loop of Henle where active transport of sodium chloride happens into the medullary interstitium. Okay. Then what happens? In response to this, from the descending limb of loop of Henle, what is happening? Water is getting added. Okay. Ultimately, finally what is happening? The medullary interstitial hyperosmolarity goes to an extent of around 1200 milliosmoles per liter. By now, you should have this query. Water, the descending limb of loop of Henle, tries to maintain an osmotic equilibrium by passively diffusing water into the medullary interstitium. That is, reabsorption of water happens from the descending limb of loop of Henle. If this water stays in this place, if this water stays into the medullary interstitium, won't this water dilute the interstitial hyperosmolarity? Of course, this water, if it stays here, it is going to dilute the medullary interstitial hyperosmolarity. For that only we have the vasa recta. What is it? What is the function of vasa recta? It is the exchanger which maintains the interstitial hyperosmolarity. So, that is where the role of vasa recta comes. This water which is added in this interstitium will be immediately reabsorbed by the vasa recta. Which portion of vasa recta? The ascending limb of the vasa recta. That is the water that moves out from the descending limb of the loop of Henle will be reabsorbed. This water which moved from the descending limb of loop of Henle will be reabsorbed. Can you see here? It will be reabsorbed by the ascending limb of vasa recta. So when this water enters into the ascending limb, where it will go? It will go into the general circulation. Okay, so once it goes to general circulation, what is its use? It maintains blood volume or fluid volume. This water is used to maintain the fluid volume. Okay, now what has happened finally? It is only the solutes which is remaining in the interstitium. The water which came was immediately reabsorbed by the vasa recta. Okay, keep this in mind. With this, we will now next move on to the vasa recta. Before looking at the vasa recta, let us also see what is the role of the distal tubule and the collecting duct in excreting a concentrated urine. We already saw the role of the descending limb of loop of Henle. We saw the role of ascending limb of loop of Henle. Try to recall and tell me how will the fluid be here, the fluid that is present in this portion of the loop of Henle. So we have your descending limb of loop of Henle. So how is the fluid in the descending limb of loop of Henle? Will this fluid be, water is moving out from her. So how will it be? Hyperosmolar. Okay. Now as it ascends up, what will happen? It will become hypoosmolar. Okay. When it goes up further, look at the osmolarity of the fluid here. Look at the osmolarity fluid at the end of the ascending limb of loop of Henle. How is it? Hypoosmolar. So that means in the distal convoluted tubule, the filtrate is hypoosmolar. Okay. Why? Because even in the distal convoluted tubule, what is happening? Some amount of sodium and chloride is leaving the cell. Okay. But later, by the time it reaches the collecting duct, this is the cortical portion. This is the cortex. Okay. The interstitium is this is cortical interstitium. This is the medullary interstitium. Okay. So in the cortical interstitium, what will be the osmolarity? It will be around 300 only. Now, by the time this fluid is coming in this distal convoluted tubule, what happens? A lot of substances will get secreted into the fluid. Most important is urea is getting secreted. Okay, that is why from 100 now it has increased to 300 milliosmoles per liter. Okay, but at least remember that the distal convoluted tubule in the renal cortex, the fluid is dilute with the level of 100 milliosmoles per liter. Only later on, because of the secretion of urea and also the presence of ADH, the osmolarity will increase to 300. Okay. Now, we were just talking about urea. So, the medullary interstitial, how much did I say this the medullary interstitial hyperosmolarity? It is around maximum is 1200 milliosmoles per liter. In this 1200 milliosmoles per liter, this ions like sodium and chloride contributes only 50%, that is around 60 milliosmoles per liter. That means what about the remaining 600? Where is it coming from? That is coming from urea. Okay. So, urea contributes around 40 to 50% of the osmolarity. When does it contribute so much? Only when the kidney is trying to form a maximally concentrated urine. When the kidney tries to form a maximally concentrated urine, the urea contributes almost 50% of the solute load to the medullary interstitium. Again, very, very important point. Okay. Normally, a person will usually excrete around 20 to 50% of the filtered load of urea. The remaining 50% of the urea, filtered load of urea, is used to produce a concentrated urine. How it is used to produce a concentrated urine? By passively shifting the urea into the medullary interstitium. Okay, when will all this happen? What is the most important factor that is going to initiate all these things? Antidiuretic hormone. So, when antidiuretic hormone is high, large amounts of urea are passively reabsorbed from the inner medullary collecting duct into the medullary interstitium. Okay, now how does ADH cause passive reabsorption of urea into the medullary interstitium with the help of specific transporters called as urea transporter. We have something called as urea transporters. We have 4 different types of urea transporters, UTA 1, 2, 3 and 4. Among that, very important of interest for us is the UTA1, urea transporter A1, which is activated by the antidiuretic hormone. This is again a very important MCQ question. So name the urea transporter that is influenced by antidiuretic hormone. The name of the urea transporter is UTA1, urea transporter A1. Okay. So now let us look at the recirculation of urea, or the role of urea contributing to the medullary interstitial hyperosmolarity. So throughout this lecture, I would have told this word medullary interstitial hyperosmolarity so many times. By now, it should be very crystal clear to you that to produce concentrated urine, the countercurrent mechanism to be established, most important is establishing a medullary interstitium which is having a lot of solute. So for that, solutes are transferred from the tubular fluid, different portions of the renal tubule, from the descending limb of loop of Henle, from the ascending limb of loop of Henle, even the DCT, from the collecting duct, different portions, solutes are getting added. Okay, as it is getting added, in the interstitial osmolarity keeps on increasing. So the contribution for the interstitial hyperosmolarity mainly comes from 2 ions, that is from sodium chloride and from urea. Urea mainly in the presence of ADH, that is when you want to conserve water. Okay, otherwise it is mainly your sodium chloride. Now what happens to this urea? What is urea? Urea is basically a metabolic waste products produced as a result of the metabolism of proteins, right? Now this urea is completely filtered at the glomerulus. If you look at the recirculation of urea, this can come as a separate 5-mark question, recirculation of urea. So if you look at urea, this is urea, okay. So all the urea that is filtered, it is completely filtered at the glomerulus, and then it enters the proximal convoluted tubule. In the proximal convoluted tubule, around 40 to 50% of the urea is reabsorbed. Okay, reabsorption of around 40 to 50% of the urea. So the urea that was 100% filtered at the glomerulus, 50% is reabsorbed in the proximal convoluted tubule. Then after getting reabsorbed in the proximal convoluted tubule, the urea travels down into the descending limb of the loop of Henle. Okay, now when it comes to the descending limb of loop of Henle, just look, the concentration of the urea is increasing. Actually 50% of the urea was reabsorbed. So what is present in the descending limb of loop of Henle is only 50%. That is why it is written as 50% is remaining. When it says 50% remaining means from in the PCT 50% is reabsorbed from this 100. So now we have only 50% remaining. This 50% remaining, if you see, the number keeps on increasing, the concentration keeps on increasing. There are 2 factors which contributes to this increase in concentration. What can happen? One is when you add more urea, the concentration of urea will increase. Another way is if you remove the fluid, then what will happen? From the solution that is having urea, if you remove the fluid, what will happen? Then also the concentration will increase. Actually, both the things are happening here. Okay, what is happening here is, we know the distal convoluted tubule is permeable to water, and it is at this portion, what is happening? Reabsorption of water happens. That is, from the fluid in the descending limb of loop of Henle, water moves into the medullary interstitium. Okay, so this area becomes concentrated. That is one reason why the number is increasing, the concentration of urea is increasing. The second, another important reason, already we told, no, the factors, if you remember those 5 factors which we saw initially. Passive diffusion of urea. Passive diffusion of urea. What is the name of the transporter? UTA1. In the presence of ADH, ADS causes passive diffusion of urea. With the help of the urea transporter UTA1, what is happening? It is, this urea is reaching the medullary interstitium. From the medullary interstitium, this urea is entering into the descending limb of loop of Henle, and also the tip of the loop of Henle, and also the ascending limb of loop of Henle. So why it is entering only these portions? Only because these regions are permeable to urea. Permeable to urea. Okay. All these thickened portions are impermeable to urea. That is why urea from this medullary interstitium, it doesn't enter any of these area. What are these areas? The thick ascending limb of loop of Henle, the distal convoluted tubule, the outer cortical collecting duct, and outer medullary collecting duct. All this region, okay, all these regions what I just mentioned, they are impermeable to urea. So urea doesn't enter this region. Okay. So this is called as recirculation of urea. This is how urea recirculates. See, such an human body, human kidney is so specialized. Urea is a metabolic waste product. It excretes around 50%. In the previous slide I told you, usually 20 to 50% of the filtered load is excreted. The remaining waste product, that 50%, the kidneys use it, even the waste, it is recycling. It is reusing this waste to increase its interstitial hyperosmolarity. Okay. So that was with respect to recirculation of urea. So what have we seen so far? We have seen the countercurrent multiplier. What are the 5 important factors that aid in the countercurrent multiplication? And what are the 7 important steps through which this countercurrent multiplication takes place? Okay, and then finally we also saw the role of the distal convoluted tubule and the role of the collecting duct, especially the role of urea in increasing the medullary interstitial hyperosmolarity. Any of this can come as a separate short note, and you can use the notes from these PPT slides to write your answers. Now let us move on to the function of countercurrent exchanger. Okay, so now we have established a hyperosmolar medullary interstitium. Not only it is hyperosmolar, but also a gradient is established. Now, if you don't maintain this gradient, what will happen? Immediately it will be washed away, because we know the blood flow of the kidney is also quite high. How much is the GFR? 180 liters per day, 125 ml per minute. So now what will happen is, if we don't have a mechanism that is going to conserve this hyperosmolarity, very easily it will be washed away. Okay, but to prevent this only, we have the vasa recta. What is the vasa recta doing? It preserves the hyperosmolarity. Please be clear. Vasa recta preserves the hyperosmolarity of the renal medulla. Now they can ask you, what are all the ways by which vasa recta preserves the medullary hyperosmolarity? That was already established by the loop of Henle. 2 things. If you look at the blood flow in the medulla, though kidneys have very high blood flow, the moment you look at the blood flow of the medulla, it is less than 5%. That means 95% of the renal blood flow goes only to the cortex. 95% of the renal blood flow goes only to the cortex. In the inner medullary interstitial region, it accounts for less than 5% of the renal blood flow. This renal blood flow is just enough for metabolic needs of the medulla. Okay, do you understand? This blood flow is just enough to meet the metabolic needs of the medulla. What is the metabolic needs? Supply of nutrients and oxygen. Only that much of blood is coming into the medulla so that the medullary cells are alive and functioning. Okay, so to meet the metabolic needs of the medulla, we need only less than 5% of the blood. So the blood flow to the medullary interstitium is very, very low. When the blood flow is less, then the interstitial hyperosmolarity will be maintained. It will not be washed away very easily. That is why the blood flow is less. The second thing is the vasa recta, okay, which acts as a countercurrent exchanger, minimizing this washout. Okay. Now, if you see, what is this vasa recta actually? So, what is this portion? This is the afferent arteriole. The afferent arteriole enters the glomerulus, branches multiply, it forms stuff capillaries, and when it leaves the glomerulus, it leaves as efferent arteriole, right? Now, this efferent arteriole, what does it do? This efferent arteriole is again branching out excessively and forms the peritubular capillary. That is, the efferent arteriole forms the peritubular capillaries. What is the importance of capillaries? Capillaries are the seat of exchange. All those things that is reabsorbed, where will it go? It has to go into the bloodstream. right? So for that you need the capillary. So now the nephron, any portion of the nephron is surrounded, very closely surrounded by capillaries. So the efferent arteriole forms the peritubular capillaries, which is surrounding the proximal convoluted tubule, portion of the distal convoluted tubule, every part of the nephron that is present in the cortex. Okay, let us think this to be the cortex. Now, the same peritubular capillaries, when it reaches the renal medulla, the same peritubular capillary, when it reaches the medulla, it is going to take the shape and form exactly like that of the loop of Henle. It is going to closely follow the loop of Henle. That means it is also going to have the same U-shaped structure. The movement of fluid is going to be counter to. Okay, all those things are going to happen. Now, this peritubular capillary is no more called as peritubular capillaries. We are calling it by a special name called as vasa recta. That means the capillaries that surround the loop of Henle in the medullary interstitium is called as vasa recta. I hope now you are very clear about what is vasa recta. What is vasa recta? It is the capillary that surrounds the loop of Henle in the medullary interstitium. Again, another thing what I clearly told you, you already know. This is your vasa recta. Okay, this red color structure is the vasa recta. This yellow color structure is the loop of Henle. And if you see, look at the direction. This is the ascending limb of vasa recta. This is the descending limb of vasa recta. Okay, so this is the ascending limb. This is the descending limb. Now look at the loop of Henle. This is the descending limb of loop of Henle. That means the descending limb of loop of Henle is in close approximation to the ascending limb of vasa recta. Then the ascending limb of loop of Henle is in close approximation or in close contact with the descending limb of vasa recta. Please remember this concept clearly. Okay. Now what did I say? What is the function of vasa recta? Vasa recta is going to maintain the medullary interstitial hyperosmolarity. How do you think it can maintain the medullary interstitial hyperosmolarity? One, by retaining the solute. Retaining the solutes and reabsorbing the reabsorption of fluid, solvent. Okay, that is water. Okay. So what is it going? First, let us all, how will we start? We will start from the descending limb and then we will move to the ascending limb. Okay. So when you look at the descending limb of the vasa recta, look at the medullary interstitium which is very close to the descending limb of vasa recta. How will the osmolarity of this interstitium will be? It is going to be high. Why the medullary interstitial osmolarity is going to be high? Because we know from the ascending limb of loop of Henle, solutes are emptied into the medullary interstitium. Now, this solute from the medullary interstitium is actually entering into this blood vessel, that is the vasa recta. Okay, fine. So, the descending limb of vasa recta takes the solute. Okay, at the same time, it is also leaving water. Okay, it is taking the solute and at the same time, water is leaving the vasa recta. Okay. Now the vasa, from the vasa recta, the water will enter into the ascending limb of vasa recta. Not only that, already if you remember I told you, from the descending limb of loop of Henle, what is happening? Water is coming out into the medullary interstitium. And I told if this water stays into the medullary interstitium, what will happen? It will dilute the interstitial osmolarity. We will not be able to establish the interstitial osmolarity. Now what will happen? As the fluid descends down from the descending limb of vasa recta. Please see, from 300 the number is increasing to 500, 600, 900, 1200. Why do you think it is increasing? Because from the medullary interstitium, what is happening? The solute that came out from the ascending limb of vasa recta is entering into the loop of, from the ascending limb of loop of Henle is entering into the descending limb of vasa recta. That is why the solute load is increasing. But do you want the solute to move from interstitium into the vasa recta? That means what? It is not beneficial, right? So whatever solute it has taken back, it has to return it. When will it return? When the blood is moving up. That is, when the blood is moving up into the ascending limb of vasa recta, all those solutes it took, it will give back. All the solutes that it took in the descending portion, it will give back. That means what is happening? The solutes are getting recirculated. The solutes are getting recirculated between the descending and ascending limb. So ultimately the solutes remain in the medullary interstitium. What about the water? The water from the medullary interstitium enters into the ascending limb of vasa recta, and as I told you, this ascending limb of vasa recta is contributing the fluid to the general circulation so that the body fluid volume is maintained. Now I hope you have understood very clearly the role of vasa recta in maintaining the medullary interstitial hyperosmolarity. What is it doing? It is basically recirculating the solutes and reabsorbing the water and taking it into the general circulation. That's all. So that completes our discussion on the countercurrent multiplier and countercurrent exchanger. Okay. So now let us move on to discuss some factors that influence the countercurrent mechanism. Okay. So now that is how through the countercurrent multiplier and countercurrent exchanger, the medullary interstitium is hyperosmolar. Okay. And the third point I told you in the starting was the role of the collecting duct as osmotic equilibrating device, right? So what does the collecting duct do? So we said the third point I told was collecting duct osmotic equilibrating device. So basically what it is doing, passive movement of urea. Passive movement of urea will be followed by movement of water as well. Okay. Why? Because when the solute concentration is increasing in this portion, that is in the medullary interstitium, what will happen? Urea is a osmotically active particle. So when it is moving, it is going to drag water along with it. So that equal amount of solute and fluid are moving from the collecting duct into the medullary interstitium. That is why the medullary interstitium is called as the osmotic equilibrating device. Another important point what I want you to remember here regarding the medullary interstitium is the cortical portion of the medullary interstitium. The cortical portion of the medullary interstitium is more permeable to water. Under the influence of antidiuretic hormone, it is the cortical portion of the medullary interstitium which reabsorbs more water compared to the inner medullary region. Why? This is again a protective mechanism so that the inner medullary region doesn't become diluted. So these 2 points also I want you to remember with regards to the collecting duct as a osmotic equilibrating device. So we have had a detailed discussion on countercurrent multiplier and countercurrent exchanger. How loop of Henle multiplies the medullary interstitial osmolarity, how the vasa recta maintains that interstitial hyperosmolarity. Now let us look at the factors that is influencing this countercurrent mechanism. First factor is the number of nephrons. Okay, greater the number of nephrons, more effective is going to be the countercurrent mechanism. Second, very important is the length of the loop of Henle. Compared to the cortical nephron, the juxtamedullary nephrons are going to have longer loop of Henle. The more longer the length loop of Henle, greater will be the gradient that is going to be established. Okay, that is one factor. Second is proximation of the tubule. The more close the tubules are to each other, easier will be the movement of solute and solvent. So that is directly proportional. The proximation more closer the tubules, better will be the countercurrent mechanism. Rate of flow in the collecting duct. Okay, if the rate of flow is very rapid, then what will happen? There won't be enough time for urea to passively move into the interstitium. So the rate of flow is inversely proportional. That is why I told the rate of blood flow is less than 5% of the renal blood flow enters the medullary region. So the blood flow is very slow. When the rate of blood flow is low, then adequate time will be there for the urea to come from the medullary collecting duct into the medullary interstitium. Okay, same thing only here. The rate of renal blood flow is inversely proportional. Greater the renal blood flow, lesser will be the gradient that will be established. And then finally the availability of urea. When you have more urea, more interstitial hyperosmolarity. Such a situation happens when we take a high protein meal. When we consume a high protein meal, the urine is going to be concentrated urine. We will produce a concentrated urine. Why? Because when you take a high protein meal, what is the end product of protein metabolism? Ammonia. which is converted to urea in the liver. So that urea only enters into your collecting duct, right? Now more protein, more urea, better concentration of the urine. Okay, better concentrating ability of the kidney to the urine. Okay. Now this is a small table which is telling us the effects of concentration and dilution on the volume and osmolarity. Already we saw the same thing, it is more like a revision. Effects of concentration. This is a very simple tabular column. What are we saying? What is the effect of concentration and dilution on the volume and the osmolarity of the urine? Please note that the osmotic load excreted is the same. That means the solute excretion remains stable. Okay, the solute excretion remains stable. Normally, at least a minimum of 600 milliosmoles per day of solute needs to be excreted. So here the solute load that is being excreted is 700 milliosmoles per day. That is stable. Now let us look at the effect of concentration and dilution. First we will start with a urine that is formed isotonic to that of plasma. When will we form an isotonic urine to that of plasma? When the GFR is around 125 ml per minute. That is normally. Normally, how much amount of water is reabsorbed? 98.7% of water is reabsorbed. So we are able to form around 1.4 to 2 liters of urine per day. The osmolarity is around 200 milliosmoles per kg of water. In such a situation, there is no net gain or loss of solute. Whereas, think of a condition where vasopressin is there, ADH is present, in maximum ADH is present. Then what will happen? The GFR is around 125 ml per minute, whereas the percentage of water that is reabsorbed in the presence of antidiuretic hormone, 99.7% of water is reabsorbed. Then how much of urine will be formed? Only 0.5 liters of urine is formed. And if you look at the osmolarity, it is around 1400 milliosmoles per kg. That means how much is the gain? We are gaining around 1.9. Okay. Just the opposite happens in the absence of vasopressin. That is, in diabetic insipidus, the percentage of the filtered water that is reabsorbed is very, very less. 87.1% of water is only reabsorbed. That means 23 liters of water is excreted per day. When you have such dilute urine, the osmolality is only around 30 milliosmoles per kg. So the loss is around 20.9. Okay. This is the effect of concentration and dilution on the volume and osmolarity of the urine. Okay. Now, let us look at what do you mean by dilute urine, what do you mean by concentrated urine. What is the mechanism that helps in forming a dilute urine? When you say the kidney is forming a dilute urine, that means the urine is hypotonic. Okay. That means the osmolarity is less than that of the blood. When will such a dilute urine be produced? What are all the conditions where dilute urine is formed? When the antidiuretic hormone levels are very less, as seen in central diabetes insipidus, or the antidiuretic hormone like in nephrogenic diabetes insipidus, where the antidiuretic hormone is present but it is ineffective. In such scenarios, the urine will be dilute. The opposite of this is the production of concentrated urine. So now compare these two pictures and see. So what you see A, this is A, this is B. B is a normal condition. Here we are producing a dilute urine. Okay. So here please compare this region. What is this portion? This is the distal convoluted tubule and then the collecting duct. So if you see, normally also the fluid that enters the distal convoluted tubule is hypotonic. Okay. Here also it is hypotonic. Okay. Then what is happening? As the fluid moves towards the collecting duct, what is happening? The osmolarity increases to 300 milliosmoles per liter, right? I told you what is happening here in the distal convoluted tubule, what is getting added? Solutes are getting secreted. Okay. And also in the presence of antidiuretic hormone, what is happening? Water is reabsorbed. So when water is reabsorbed, what is happening? When water is reabsorbed into the medullary interstitium, what will happening? This interstitial osmolarity increases from 100 to 300. As you keep going down, it keeps on increasing, and at the tip it is around 1200, because as you keep going down, what happens? Not only water leaves, even sodium chloride, urea, all the solutes are also leaving. So the interstitium is hyperosmolar, and we also know the proportion of water that is reabsorbed is so much less compared to the proportion of solute that is reabsorbed into the medullary interstitium. This happens normally. In case we are forming a dilute urine, what is happening? You see, in the absence of antidiuretic hormone is not there. No antidiuretic hormone. Then what is happening? The osmolality of the fluid, which was hypotonic 100, as it keeps coming down, what is happening? It keeps on decreasing. Why it keeps on decreasing? Water is not getting reabsorbed. Why it decreases? Because no reabsorption of water. No reabsorption of water into medullary interstitium. This is important. Water is not getting reabsorbed from the lumen of the collecting duct into the medullary interstitium. Plus, solutes leave the collecting duct fluid. Okay, so from the collecting duct, solutes, see, sodium chloride is leaving. Okay, even urea will leave, but water is not leaving. So now what is happening? It is becoming, solutes are also leaving, so it is becoming more and more diluted. Okay, this is the effect of absence of antidiuretic hormone. That is, here we are forming a dilute urine, here you are forming a normal urine. Now I think you are able to understand the difference between how the kidneys are diluting the urine. Okay, now the opposite scenario. What will happen if we want to form a concentrated urine? Very, very simple. What is happening? If you want a concentrated urine, that means the osmolarity should be more than that of the plasma. When will it happen? When the antidiuretic hormone levels is high, we are going to form a concentrated urine. Okay. So what are all those conditions where the circulating antidiuretic hormone levels are high? In case of water deprivation, that is dehydration. When a person is dehydrated. Then when a person has lost body fluid in the form of hemorrhage, and syndrome of inappropriate antidiuretic hormone secretion, where inappropriately high levels of antidiuretic hormones are secreted. In all these scenarios, what are we going to see? We are going to have a greater level of antidiuretic hormone. And you know, if antidiuretic hormone is there, water will be reabsorbed from the collecting duct. So ultimately, the solute load is going to be high. Okay. So that is why and along with this, it also augments the urea cycling. So all this factor put together will increase the osmolality of the urine and decrease the volume of urine that is formed. Okay. So this is finally, before we end, let us just briefly look at the segmental changes that happens in the tubular fluid during the formation of urine. Very simple. This is because they ask in your Viva and MCQ. That is why I am specially discussing it here. What do you mean by segmental changes? Basically, how is the osmolality of the fluid in the tubule? We are not talking about the medullary interstitium, we are talking about the filtrate. Okay. So, in the proximal convoluted tubule, how is it? It is all 300, it is 300 here. So, the fluid in the PCT is isotonic to that of the medullary interstitium. Now, when you are looking at the limb of loop of Henle, look at this number. Since it is losing water, how is the fluid? Hypertonic. And keep looking at it. From 400, it increases to 600, then to 800, 1000. Where do you see the maximum hyperosmolarity? Maximum hyperosmolarity or maximum hypertonicity of the fluid in the loop of Henle is at the tip of the loop of Henle. So the tip of the loop of Henle is the portion where the tubular fluid is having maximum hypertonicity. Now, after that, as it ascends up, what is happening? It has become more and more hypotonic. So, in the ascending limb of the loop of Henle, the filtrate is hypotonic. Now, as you keep moving up, what is this? DCT. Even in the distal convoluted tubule, how is it? Hypotonic. Okay. Now we are coming to the, what is this region? Collecting duct. Okay. In the collecting duct, in the cortical collecting duct, it is isotonic. Again, in the cortical collecting duct, it is isotonic. In the presence of antidiuretic hormone, here we have to be very specific whether antidiuretic hormone is present or not. When antidiuretic hormone is present, what is happening? It is again becoming hypertonic. So, in the presence of antidiuretic hormone, the fluid in the medullary collecting duct is hypertonic. So, now you know segment specific changes in the tubular fluid. In the PCT, how is it? In the descending limb, in the ascending limb, wherever they ask you, you will be able to answer it very, very clearly. Okay. Now, what are the other systems in which we can see a countercurrent mechanism? Countercurrent mechanism takes place for heat exchange between the arteries and veins in the limbs. Then it is also present in the intestinal villi. But the countercurrent that is present in the intestinal villi is disadvantageous because of this countercurrent flow, the nutrients are getting dissipated. They are not reabsorbed properly. Okay. Then in the brain also, the arterial blood temperature is regulated. There also we have the countercurrent mechanism. And finally, in the testis, countercurrent mechanism is seen in the pampliform plexus of veins, where it helps in maintaining the temperature and also helps in maintaining the high levels of testosterone. So, these are the other areas in the human body where we find similar countercurrent mechanism. Okay. Now, the test, what are all the tests that is available to assess the renal diluting and concentrating ability? Which is the most important index? In the starting itself, I told you, measurement of urine osmolality. If the osmolality is less, dilute urine. If the osmolality is more, concentrated urine. Same thing applies to specific gravity, directly proportional. Greater the specific gravity, more concentrated. Lesser the specific gravity, less concentrated. What is a normal specific gravity? 1.005 to 1.030. Okay, this is the normal value. Then urine concentration test and urine dilution test. What we do in urine concentration test, we will limit the fluid intake. We will limit fluid intake. So, when we limit fluid intake, we will check the concentrating ability of the kidney. In urine dilution test, drink fluid. Okay, you are going to increase the fluid intake and then we will check how much of dilute urine the kidney is able to excrete. And finally, free water clearance. What do we mean by free water clearance? We are going to measure the amount of water that is free, that is excreted, which is completely free of solute. Okay. If the free water clearance is positive, that means we are excreting a dilute urine. If the free water clearance is negative, the person is excreting a concentrated urine. Okay. So, these are the various methods used to assess the renal diluting and concentrating ability. So, finally, before moving into our clinical case scenarios and multiple choice question, a brief look at the different applied aspects. Okay. First, let us look at primary polydipsia. In this primary polydipsia, what happens is the person keeps on drinking a lot of water. So, when you drink a lot of water, what will happen to the osmolarity of the plasma and the concentration of sodium? This excess of water intake is going to dilute the blood. So, the solute load and osmolarity will be decreased, right? And how will be the urine osmolarity? Already plasma is hypotonic. So, the urine osmolarity is also going to be hypoosmotic because they are drinking lot of fluid, not correcting the electrolytes. Okay. So, then what is going to happen? More amount of fluid, so more will be the rate of urine flow. So, we are forming a dilute urine. When you form a dilute urine, the free water clearance will be positive. In primary polydipsia, the serum antidiuretic hormone levels are decreased. Okay. Next, we have next condition, central diabetes insipidus. We have nephrogenic diabetes insipidus and central diabetes insipidus. What is the difference? In central diabetes insipidus, antidiuretic hormone level itself is less. In nephrogenic diabetes insipidus, it is either normal or it can even be increased. Then what is the difference between these two? In nephrogenic, the kidneys are not effectively able to utilize the antidiuretic hormone. In central, antidiuretic hormone is not at all available. So if ADH is not there, what will happen? Definitely, what is the kidney going to do? It is not going to conserve water. So if it is not going to conserve water, what will happen? It is going to excrete a large amount of water. So the osmolality, hypoosmotic urine will be formed. Urine rate will be high. Definitely free water clearance is positive. But what we have to remember here is, since the body is losing a lot of water, the cells become dehydrated. Okay. So because of dehydration, what is happening? The sodium load and the osmolality of the plasma increases. Okay. Now I hope you understood why this is increased. In nephrogenic diabetes insipidus, everything is the same. Only difference in nephrogenic diabetes insipidus, the ADH levels may be normal or even increase. The kidneys are not able to utilize it, so it has a similar picture of diabetes insipidus. Okay. Then in water deprivation, what do we mean by water deprivation? That is dehydration. When a person is dehydrated, what is happening? Whenever a person is dehydrated, immediately antidiuretic hormone will be released. Okay, so antidiuretic hormone levels are increased. But in dehydration, what will happen to the plasma osmolarity? Of course, the plasma osmolarity is depending upon the degree of dehydration. It can be high or sometimes normal. The urine, now already water is not there, ADH has been released to conserve water. So definitely the osmolarity of the urine will be high, hyperosmolar urine. The volume of urine is going to be low. When you pass a low hyperosmolar urine, the free water clearance is negative. Okay. Finally, let us discuss syndrome of inappropriate ADH secretion. What happens here? Inappropriately high amount of ADH is secreted. Because inappropriately high amount of ADH is secreted, what will happen? Lot of water is going to get reabsorbed. Inappropriately high amounts of fluid is getting reabsorbed. Okay, when high amounts of fluid is getting reabsorbed, what will happen? The solutes are all getting diluted. So if you look at the plasma osmolarity and the serum sodium concentration, because of the increased fluid reabsorption, it decreases. The rest of the things remains the same, like that of dehydration. That is, kidney, because of excessive ADH, is excreting a low volume urine with high osmolarity. Thereby, the free water clearance also is negative. Okay. Now we have completely in-depth discussion of countercurrent mechanism of the kidneys have been done. Let us recall whatever we have learnt by answering these questions. If you are able to answer these questions, that means you have understood this concept well. So we have the clinical, we know in your examination with the current CBME curriculum, we have some clinical reasoning based questions, short notes, long question and MCQs. We have already discussed all the topics for short notes and MCQs, right? And long question as well. Now let us look at a few clinical reasoning questions pertaining to this topic. The first question is, a patient with chronic kidney disease presents with an inability to concentrate the urine. So we have a patient with some kidney disorder. Because of the kidney disease, he is not concentrating the urine. What are they asking you? Explain how the disruption of this countercurrent mechanism in the nephron could lead to this condition. Okay, now these clinical reasoning questions are all either for 2 or 3 marks. So they want you to give a physiological response or the physiological phenomenon in a very crisp manner. We don't expect you to write pages for this type of question. Okay, so what is the question here? In patients of chronic kidney disease, how is the countercurrent mechanism disrupted? That much is what they are asking. So what will you write? We all know countercurrent mechanism is important for establishing a hyperosmotic medullary interstitial gradient. Now, because of the chronic kidney disease, what happens? There is structural damage to the nephron or there is reduced renal perfusion. Any of these things is going to affect or disrupt the medullary gradient establishment, thus reducing the kidney's ability to concentrate the urine. That's all. Simple 2 to 3 line answer is what we expect. Okay. Second question. We have a patient on dopamine infusion. Okay. Why this patient is given dopamine infusion? Because he is having shock. To improve the urine output, patients of shock go into renal failure. So, and complete shutdown, renal shutdown. Zero urine will be produced. So, to improve his urine output, this patient is given dopamine. Already they have clearly mentioned here, dopamine is a vasodilator. What are they asking you? Explain the physiological basis on how a low dose dopamine affects the renal perfusion and sodium handling. Okay, they're basically asking the physiological basis of the action of dopamine. Okay, the clue is already given in the question. Dopamine is a vasodilator. When it causes vasodilation, what will happen to the renal blood flow? Renal blood flow in low doses, dopamine is causing vasodilation. Vasodilation increases the renal blood flow. If renal blood flow increases, GFR increases. Okay, and because of this, what will happen? The sodium excreting capacity is also increasing. When the GFR increases, what will happen? The urine output also increases. Okay, not only that, additionally, this dopamine inhibits the sodium reabsorption from the proximal tubule. So that is going to cause natriuresis, excretion of sodium. Whenever natriuresis happen, it is followed by diuresis as well. So collectively, that is how dopamine is increasing the urine output. Okay, next question. We have a patient who presents with polyuria and hypotension. We have a patient who is presenting with polyuria and hypotension. Lot of water is excreted from the body. So the patient is going into shock. The blood pressure decreases, hypotension. How could impaired antidiuretic hormone secretion affect the renal countercurrent mechanism? So basically they are asking the role of ADH in countercurrent system. What is it? What is ADH basically doing? It facilitates water reabsorption. How it facilitates water reabsorption? Basically from where? From the collecting ducts. What it is doing? How water is reabsorbed in the presence of ADH? Aquaporin 2 channels are upregulated and they reabsorb water. Okay. Now, when ADH level is deficient, as in case of central diabetes insipidus, or ADH is normal but the kidneys are not able to respond to this ADH. In any of this condition, what is going to happen? ADH is not available. The person is excreting lot of urine, a lot of water in the form of urine, leading to hypotension. Okay. Now, very few MCQs before closing the topic. Which part of the nephron is primarily responsible for establishing the medullary concentration gradient? Okay. Which part of the nephron? Medullary concentration gradient is established by your loop of Henle. Right? Now, what is the main role of urea in recycling the kidney? Is it to improve the glucose reabsorption, or is it to maintain potassium homeostasis, or to enhance the medullary osmotic gradient, or to regulate renin secretion? Yes, it's a direct question. It is to enhance the medullary osmotic gradient. Urea enhances how much? 50% is contributed by urea. Okay. In the absence of antidiuretic hormone, what happens to the urine osmolality? Just now we saw, if antidiuretic hormone is not there, the person is going to excrete a dilute urine. So the osmolality is going to decrease significantly. Okay. Next question. Which nephron segment has the highest tubular fluid osmolarity under normal hydration? So which portion of the nephron segment has the highest tubular fluid osmolarity? Okay. What is it? Direct answer. It is the tip of the loop of Henle. You remember I discussed it is a tip of the loop of Henle. How much is it at the tip? Maximum it can go to 200 milliosmoles per liter. Okay. Now, what happens to the osmolarity of the tubular fluid as it moves through the ascending limb of loop of Henle? He is asking. So, this is the ascending limb of loop of Henle. This is the descending limb of loop of Henle. So, when it moves down the descending limb, what is happening? From 300, it becomes 800, 1000, 1200. Then as it attends up, it keeps reducing 1000, then 800, 600. So, what is happening here? What is happening to the osmolality of the tubular fluid? If you see in the ascending limb of loop of Henle, is it increasing or decreasing? It is decreasing. It decreases significantly. Okay. Which nephron segment is primary site of urea reabsorption? Which is the primary site of urea reabsorption? It is, what is it? It is a medullary collecting duct. This is the major site of urea reabsorption. Now, next question. In which part of the nephron does urea concentration increase the most? Which is the part? Just now we discussed. It is the collecting duct. To be more specific, it is the medullary collecting duct. Okay. So that finishes our discussion on countercurrent mechanism. We had a detailed, in-depth discussion of countercurrent mechanism, and this is the last slide, which is showing you the summary of all those events we saw. I would highly recommend you to go through the summary after reading the topic completely, so that you will be able to recall the key events carefully. Okay. Thank you and all the very best.